The replication of a chromosome begins at particular sites called origins of replication, short stretches of DNA having specific sequences of nucleotides. Proteins that initiate replication attach to DNA, separating the two strands and opening up a replication bubble. Multiple replication bubbles form and eventually fuse. At the end of each replication bubble is a replication fork, a Y-shaped region where the parental strands of DNA are being unwound. Firstly, helicases are enzymes that untwist the double helix at replication forks, separating strands and making them available as template strands. Single-stranded binding proteins bind to unpaired DNA strands, keeping them from repairing. Topoisomerase breaks, swivels, and rejoins the parental DNA ahead of the replication fork, relieving strain caused by unwinding. The enzyme primase synthesizes RNA primers using parental DNA as a template. RNA primase starts a complementary RNA chain from a single RNA nucleotide, adding more RNA nucleotides one at a time until the primer is about 5 to 10 nucleotides long. The new DNA strand will start from the 3' end of the RNA primer.
Elongation process is different in the 5' to 3' template and the 3' to 5' template. The 5' to 3' end is called a leading strand because DNA polymerase III remains in the replication fork on that template strand and continuously adds nucleotides to the complementary strand as fork progresses. The 3' to 5' strand, called the lagging strand because it is synthesized discontinuously as a series of segments known as Okazaki fragments form. Primase places RNA primers to the strand. DNA polymerase III adds DNA nucleotides to the primer, forming the first Okazaki fragment. After reaching the next RNA primer to the right, DNA polymerase III detaches. DNA polymerase I replaces the RNA with DNA, adding nucleotides to the 3' end of fragment and earlier of fragment 1. DNA ligase forms a bond between the newest DNA and the DNA of fragment 1, joining the sugar phosphate backbones of the Okazaki fragments. The lagging strand in this region is now complete.
Transcription is the process of making an RNA copy of a gene sequence. This copy, called the messenger RNA molecule, leaves the cell nucleus and enters the cytoplasm, where it directs the synthesis of the protein which it encodes. Transcription is made up of 3 stages: initiation, elongation, and termination. A eukaryotic promoter commonly includes a title box and nucleotide sequence containing TATA about 25 nucleotides upstream from the transcriptional start point. Several transcription factors, one recognizing the title box, must bind to the DNA before RNA polymerase II can bind in the correct position and orientation. Additional transcription factors bind to the DNA along with RNA polymerase II, forming the transcription initiation complex. RNA polymerase II then unwinds the DNA double helix, and RNA synthesis begins at the start point on the template strand.
The process of transcription produces pre-mRNA, which is then edited to produce the mRNA that leaves the nucleus and is later translated into a protein. During this process, a 5' end and a poly-A tail are attached to both ends of the mRNA in order to facilitate the export of the mRNA from the nucleus, protect the mRNA from degradation due to enzymes, and help ribosomes attach to the 5' end once the mRNA reaches the cytoplasm.
Then farther along in the process of RNA editing, splicing occurs where non-coding segments of nucleic acid between coding regions called introns are removed with the help of a large complex of proteins and small RNAs called a spliceosome, leaving only exons, the coding region of the nucleic acid.
Translation begins with initiation, in which the tRNA forms a hydrogen bond with the AUG start codon. After the mRNA, initiator tRNA, and small ribosomal subunit are united, a large ribosomal subunit attaches to form the translation initiation complex by using proteins called initiation factors. The next stage, elongation, occurs in 3 steps: codon recognition, peptide bond formation, and translocation. During codon recognition, the anticodon of the tRNA binds to the corresponding mRNA codon using the energy from hydrolyzed GTP to increase accuracy and efficiency. Then the large ribosomal subunit catalyzes the formation of a peptide bond between the amino group of the new amino acid in the A site and the carboxyl at the end of the growing polypeptide from the tRNA in the P site. The formation of the peptide bond removes the polypeptide in the P site and attaches it to the amino acid on the tRNA located in the
The
A
last
site.
step of elongation is called translocation, in which the ribosome uses energy to translocate the tRNA in the A site to the P site, while the empty tRNA in the P site is removed to the E site to be released. The mRNA moves along with its bound tRNAs to bring the next codon to the A site, and the cycle repeats. The last stage of translation, termination, occurs when one of the stop codons, UAA, UGA, or UAG, on the mRNA reaches the A site and the process of elongation is stopped. Gene expression in bacteria can be divided into two categories, negative regulation and positive regulation. In negative regulation, there are repressible operons and inducible operons. Repressible operons usually have their transcription on, but transcription can be inhibited when a specific small molecule binds allosterically to a regulatory protein. Inducible operons, in contrast, are usually off, but can be stimulated when a specific small molecule interacts with a regulatory protein. In positive regulation in bacteria, a regulatory protein known as catabolite activator protein binds to DNA and stimulates transcription of a gene by increasing the affinity of RNA polymerase binding to the promoter, which increases the rate of transcription.